how's it going everybody? John here, and in 2007, Gautam Dantas, who at the time was a postdoctoral researcher in a lab at Harvard University, was doing some research that involved bacteria that were put in two different dishes. Now to one dish, he added an antibiotic to a dish that's got bacteria in it, okay? And to the other dish, he added plant waste as a source of food. After 1 week, he finds that the bacteria that were given the plant matter as food had only grown a little bit, while the bacteria that had antibiotic added to them were having a party. They were having a blast. They used the antibiotic as food. They ate the antibiotic. Now this was so unexpected to him that he actually thought that they had made some kind of mistake. Except it wasn't a mistake. This was real. Now this is very different from just regular antibiotic resistance. And to clarify, let me tell you a story. So let's say you got sick and you went to see your doctor. Your doctor diagnoses you and tells you you have an infection. And he prescribes you an antibiotic called amoxicillin. So you go with the prescription to the pharmacy, you fill your prescription, you take the medication home and you start taking it. You take it for several days and you feel no improvement whatsoever. You give your doctor's office a call and say, hey, this isn't working. Can you see me again? Can you prescribe me something different? And the doctor says, sure, come on in and let me see you. You go back into the doctor, he gives you another checkup, and he prescribes you another antibiotic and tells you, I'm going to send the prescription to the pharmacy, go to the pharmacy and pick it up. So you go back to the pharmacy, you pick the antibiotic up, and you look at the label, and the label says amoxicillin-clavulanic acid or amoxicillin-potassium clavulanate. You look at this and you think to yourself, okay, this has amoxicillin in it. I already took amoxicillin and it did nothing. Why did the doctor do that? You ask the pharmacist and the pharmacist says, well, yes, this does have amoxicillin in it, but it's not exactly the same thing. You see, the potassium clavulanate or the clavulanic acid, what that does is that it prevents the bacteria from spoiling the antibiotic so the antibiotic can kill the bacteria. So yes, the bacteria can spoil the antibiotic, and that's pretty much an example of regular antibiotic resistance. But what Dantas found in 2007 was bacteria that eats the antibiotic. Now let's be clear here, antibiotic resistance is a really serious problem. Every year, about 700,000 deaths happen because of bugs that were resistant to penicillin and other drugs that were supposed to kill them. In August of 2016, a woman in Nevada died because of an infection caused by Klebsiella bacteria that was resistant to 26 different antibiotics. Now this is a huge problem, but there's science that just might be able to help with that. Okay, now back to the bacteria that eat antibiotics. If that was a surprise to you, well, it should be. But to be frank with you, actually biologists have known that bacteria can eat antibiotics for a really long time. I mean, since the early 60s. But that knowledge just wasn't widespread. Anyway, Dantas decides to run another experiment. So he goes around and collects soil samples from different places, and that soil contains bacteria. And he tests those samples with 18 different drugs. 18 different drugs. And disturbingly, all 18 drugs were eaten up by the soil bacteria. Now this is extra disturbing because the concentrations of the drugs that were used were 50 to 100 times higher than the concentrations that regular antibiotic-resistant bacteria can tolerate. Those results about the 18 different drugs were published in a scientific journal called Science in 2008. But recently, another paper by Dantas dives into the details and explains how some soil bacteria eat the antibiotic. Dantas used penicillin, which is an antibiotic that's been around for a long time. And the penicillin family of antibiotics contains several members, one of which is amoxicillin, for example. Now, penicillin is a beta-lactam antibiotic, which basically means that part of its chemical structure is something called a beta-lactam ring. Penicillin uses this ring to weaken the cell wall of whatever it's trying to attack to kill it. Now, a regular penicillin-resistant bacteria produces an enzyme that breaks the beta-lactam ring, hence making the penicillin ineffective. But the antibiotic-eating bacteria in the soil goes farther than that. The antibiotic-eating bacteria breaks the beta-lactam ring, then it cuts it off completely from the rest of the penicillin molecule. And after it does that, the remaining part of the molecule is called phenylacetic acid. Then the bacteria eats the phenylacetic acid as food. Dantas figured out which genes in the soil bacteria give it the antibiotic eating ability, and he added those genes to the DNA of a different kind of bacteria called E. coli. And sure enough, after doing that, the E. coli bacteria were able to feast on penicillin. Now, so far, the man's done some really impressive work, but he doesn't stop there. Dantas travels to Peru to find out how much antibiotics are in wastewater treatment plants. And sure enough, he finds several highly prescribed antibiotics in the wastewater. The thing is, studies have found that there's a lot of penicillin and other drugs in wastewater that drains from pharmaceutical plants and farms. So finding antibiotics in wastewater unfortunately makes sense. Now here's a really cool part. Dantas' idea is to tinker with bacteria, to turn them into antibiotic-eating bacteria, so they can eat antibiotics in wastewater, for example, so that the water becomes antibiotic-free before it's released into the open. That way, other bacteria won't get exposed to the antibiotics, and hence, the other bacteria won't become antibiotic resistant. One more time, the man's idea is to tinker with some bacteria to make them antibiotic-eating bacteria. Use them to treat, for example, wastewater, so they eat up all of the antibiotics in the wastewater. And when that water that now contains no antibiotics is released into the open, the bacteria that it runs into later is not exposed to antibiotics. Hence, that bacteria does not become antibiotic resistant. It's a genius idea, but it's kind of risky. And here's why. You see, the thing is, bacteria, they're kind of smart. They do this thing where they share bits of their DNA with other bacteria. And those bits that they share can do smart things. So basically, if the antibiotic-eating bacteria shares the antibiotic-eating bit of DNA with other bacteria that are just regular antibiotic-resistant bacteria, they would turn into antibiotic-eating bacteria too. And we would have a catastrophe on our hands. This would kind of be the opposite of what Dantas wants to do. So instead of preventing a lot of bacteria from becoming resistant, this would help the resistant bacteria turn into antibiotic-eating bacteria. Now there is another idea, which is to have the antibiotic-eating bacteria produce an enzyme that breaks down the antibiotics. And then those enzymes would be used to break down the antibiotics in wastewater, for example. But the problem is that would be too expensive to do. It could at least be used in hospitals though, so that's a potential use. Anyway, at least there are out-of-the-box ideas. And perhaps one day they will lead to a safe and practical way to at least decrease the threat of antibiotic resistance. That's it for this video, guys. If you liked it, please like, share, and subscribe, and thank you for watching.